Hey guys, welcome back. So we're going to talk about DNA structure and DNA replication. These are the things that we've talked about from Friday all the way up until today/Thursday. So Wednesday and Thursday for our quest on Friday. So our first thing is we're going to talk about DNA and what it is and that type of stuff. So we're going to identify the substances and genes. And so some of the following experiments that allowed scientists to determine DNA was the molecule of inheritance. And so scientists knew that genetic information was carried on chromosomes. And of those chromosomes, we knew that it was made up of two things, proteins and DNA. The question was, which one of those was the source of genetic material to pass to offspring? And that was one of the things that we didn't really know. And so as we go through, these studies support one over the other. So the first major experiment was in 1928, and it was from a pneumonia outbreak. And so we had a virulent strand, or the smooth strand, and the non-virulent strand, which was the rough strand, or R strand. And so, like most medical breakthroughs that we have is following outbreaks. So in 1928, we had the pneumonia outbreak, and so we're going to use a study for this. And so from here, we had a control. We had the rough strand. We knew it was non-virulent. We knew it wasn't disease-causing, and so we injected into a mouse. The mouse lived. Then we had the smooth strand, or the virulent strand. We injected to the mouse, and the mouse died. And we had a heat-killed smooth strain, and so we put the smooth or the virulent strand into a really high amount of temperature to technically kill it or to sterilize it, and the mouse lived. And then the rough strain and the heat-killed smooth strain would combine together into the mouse, and the mouse died. And the biggest question is, why did that happen? And so Frederick Griffith, which is the guy who did this picture here, knew that heating the bacteria denatured the proteins. And because we were deciding between proteins or DNA, we knew that we denatured the proteins. There was something else that was a genetic material. And he concluded that genetic material from the virulent strand survived, which in this case was DNA, and incorporated itself into the non-virulent strand, or what we call transformation. And he concluded that proteins were not genetic material in 1928. However, in order for us to be able to kind of consider this as supported, we had to have multiple experiments. So what happened here is we took the same two strains of pneumonia, and we took the S strain cells, and then we did a fractionation of cell-free extracts into the classes of molecules. And so what we did is we put the S strain cells and we combined them with RNA, protein, DNA, lipids, and carbs from the smooth strand. And we wanted to see which one tested for transformation. Oh, let me go back. And so what we found was that RNA did not have transformation, protein did not have transformation, lipids did not have transformation, and carbs did not have transformation. The only thing that transformed into the R strand was the DNA, which means it was now a virulent strand. So, as only the non-virulent bacteria was given DNA transform, and so Avery, who did this, concluded that DNA was genetic material. And now that was in 1944, and the popular opinion at that time still favored proteins as hereditary material, which was interesting because we've already had two studies that support that it was DNA, not protein. So of course, with science, we had to do another experiment. So we had Hershey and Chase, and they studied viruses. They were virologists and they looked at viruses that infected bacteria, or what we call bacteriophages. And they tagged DNA with radioactive phosphorus, and they tagged viral proteins with radioactive sulfur. And so here's the picture of their results. And so on the left is where we tag the protein. On the right is where we tag genetic material, or DNA. So you see the bacteriophage injects the DNA into the cell. They blend their genetic material into the cell, and then from there we see which one is tagged and which one's inside and outside the cell. So we saw on the left that the sulfur detected is in the what we call the capsid or the protein covering, while on the right we saw that the phosphorus labeled was inside the cell. And so we concluded that DNA, not protein, was the genetic material. And they determined that a protective protein coat, or the capsid, was formed around the bacteriophage, but the internal DNA is what conferred its ability to produce progeny inside of a bacteria. And what that means, progeny, produce offspring. So this was in 1952. So here's the DNA molecule. Looking at it, we can kind of see, let me go back, resume. Sorry, I clicked backspace. We can kind of see the structure here. We can zoom in and out. We can look inside and see what's going on here. And so it's kind of cool in looking at the structure of DNA, which is what we're talking about next. So the structure of DNA was in 1949 with Chargaff, and he observed that the percentage of guanine was equal to the percentage of cytosine, and the percentage of adenine was equal to the percentage of thymine. And this finding was the basis of what we call the base pairing rules. And so there are two types of bases. We have purines and pyrimidines. So purines is A and G, or adenine and guanine, while our pyrimidines is cytosine, thymine, and uracil. Now uracil we're going to talk about when we talk about RNA, not necessarily right now, but it'll be good to know that because U is going to replace T when we talk about DNA to RNA. And so we should see here to the bottom we have the pyrimidines, how it's double ring. So the guanine has basically two octagons here, or a pentagon and an octagon, while the pyrimidines have just an octagon. And so then what you're going to do is you're going to look to see which one pairs with what. And so adenine and thymine pair together, and cytosine and guanine pair together. So the structure of DNA, the base pairing rules show how nucleotides always pair in DNA. So A binds with T, C binds with G. And because a pyrimidine pairs with a purine, the double helix is a uniform width. Because we have a double ring and a single ring bonding together every time. Now we have 1953, we have the structure of DNA. So within this is Watson and Crick. They came up with the model, or the double helix model of DNA, but if you read your article, not necessarily. And so the biggest thing within that is they actually got their topic, or got their last piece of data from these two, Rosalind Franklin and Maurice Wilkins. So from here, what they did was they were looking at the structure of the double helix, and they were trying to figure out what specifically was occurring and how this shape was of the double helix. And so they literally took Rosalind Franklin's picture, this picture right here to the right, with her X-ray diffraction experiments, and suggested that the DNA was a double helix with a specific width, which gave them everything they needed to know because they understood Chargaff's base pair rules. So from that, what we came to the conclusion is that we have a backbone and rungs of a ladder. So the backbone is made up of the sugar phosphate and is connected by covalent bonds, which is here. And the nucleotide bases, or in this case, the A and the T, are connected by hydrogen bonds. So A and T is 2, G and C have 3. Now the hydrogen bonds are very, very weak and they can be separated very easily, while covalent bonds are very, very strong and they're harder to pull apart. Also, the orientation of strands is another unique feature. as its anti-parallel. So for instance, the top rail to the bottom is 5' to 3', and the opposite is the top right is 3' to 5'. And so they run in opposite directions called anti-parallel. So to the picture to the left here, we have a printout of the Human Genome Project, and this is a base sequence of DNA in the order of nitrogen bases on a chain of DNA. And each person has a unique base sequence, which as a whole is called a genome. And so basically you should be able to explain and describe the structural components of DNA, and explain and describe the bonding between DNA and strands. Oh, sorry, I'll go back real quick before we talk about replication. So looking at this picture, this would be my phosphate. This would be my nitrogen base, or my, not nitrogen base, my deoxyribose sugar, and these are my nitrogen bases. So sugar, deoxyribose sugar, phosphate, covalent bonds, hydrogen bonds. So replication DNA. DNA replication is the process by which DNA is copied in a cell before the cell divides. So some important enzymes to look at in DNA replication is helicase. So helicase separates the two strands of DNA by breaking the hydrogen bonds between the strands. So anytime that you see like an opening within DNA, it's usually due to helicase breaking those hydrogen bonds. Then we have DNA polymerase, and it's going to catalyze or speed up a chemical reaction. The synthesis to make, is making of DNA by adding new nucleotides in the 5' to 3' direction. Also proofreads the new DNA sequence for errors. The ligase catalyzes the bond formation between Okazaki fragments and the lagging strand. And the Okazaki fragments are short segments of DNA formed on the lagging strand. And so you're like, well, what does this mean? So this is a replication fork. So within this replication fork, you got to be able to draw what it is. So what I have done is I've drawn one for you. Well, I was supposed to. Let me find this picture for you. All right. I believe I have added it. Yes, I want to resume it later. So let me edit real quick. Yes, I want to lose it, because I need you to be able to see this picture. It probably is the very end. Okay, it's at the very end. So we're going to go back to where we were as it loads. And so one of the biggest things is being able to draw this picture. So I'm going to skip to the very end to draw this picture. So this is the picture that we're going to draw right here. So let me zoom in a little bit for you. So see, we have my DNA polymerases here. So this is my leading strand because we're creating a whole big strand. This is going towards the helicase, as you see here at the fork. This polymerase is going away from the helicase, so this is my lagging strand. So I have two sections of DNA here. Those are my Okazaki fragments. And then I have my ligase, which is going to bind my phosphate groups from my different nucleotides together, so we can form DNA that's solid. This is my 3' end, my 5' end, my 3' end, my 5' end. And then I have RNA, RNA, and RNA. And these are primers that lead to being able to make RNA. So, DNA replication begins at an origin point. And there are multiple origin points within replication of eukaryotic cells or chromosomes. And so because we are so complex, our DNA is so wrapped around and so tight that we have to do multiple points of origin in order for it to spread out to evenly have two different strands for this replication. And so in prokaryotes, there's only one origin of replication on their circular chromosome, because a circle starts and stops at the end or at one point. And so there's only going to be one point of origin and we're going to create a whole new strand from this one helicase. So then you should be able to explain why DNA replication is more complex in eukaryotes and in bacteria, and you should be able to draw the DNA replication picture. If you guys have any questions, please let me know. I know this is kind of short and sweet, but just kind of giving you a quick little rundown of what we talked about, and I hope this helps.